Hello everyone, welcome back to our discussion on Mendelian genetics. In the last session, we discussed the four laws of Mendelian genetics, and now we are going to discuss about some special kinds of crosses. We have three of them. One is back cross, the other one is called as the test cross, and the last one is called as the reciprocal cross. So we will directly move on to this cross, which is called as the back cross. So back cross is a cross. So this can be back cross can be done both in the dihybrid as well as it can be done for both monohybrid as well as in dihybrid crosses. So this is a cross where the F1 heterozygote of a monohybrid or a dihybrid cross is crossed with either of the parent. That means it can be either the dominant parent or the recessive parent. So we'll see this clearly here. So this is what a backcross means. So backcross, so this is a monohybrid backcross. So this one is the F1 heterozygote, the heterozygote of the F1. So here what normally what we do in a monohybrid cross is that to get the F2 generation, we self fertilize F1 cross F1 and we get the Mendelian ratios. So what happens here is in a back cross, what you do is you are going to cross this F1 hybrid to either of the parent. It can be a dominant parent or a recessive parent. In either case, you get different results. So we'll see what happens when the F1 heterozygote is paired with the dominant parent. Okay, so what happens here is when so these are the two different gametes which are produced, and this is the gametes which are produced here. So the thing to be noted is that I put this particular example because if you are taking or this particular one, as we discussed earlier, it might be easy to do like this. That is, you have capital T small t cross capital T capital T. So what we said in our earlier discussion was that because these two are different, we write this gametes like this, and because this produces only a single type of gamete, you write only a capital T. This is one type of representation of a Mendelian cross, and in this you do what so here what you do is you cross with this, what you get is capital T and capital T small t. So what you get is all are tall plants. One is homozygous dominant, the other is heterozygote. Anyway, you have 100% which is tall plants. All are tall plants. So this is one type of representation of a Mendelian cross. That means you are just crossing it. If you are doing it with a Punnett square, so this square is called, as we discussed in the first class, this is called as a Punnett square. If you are doing with the Punnett square, you can take it as two different gametes because this particular individual produces or they undergo homologous chromosomes get separated. You get these two. You can take it like that also. In any case, you get the same answer. Okay, so here even though you represent or you write 4 offsprings, inside you have 4 offsprings, whatever the offsprings may be, every offspring is tall. Is that clear? That is, whatever the offsprings are produced, they are 100% tall. So that is with the other one. In this case, what will happen to when you cross the F1 hybrid or heterozygote with the recessive parent? So the same thing, you can represent it like this. You have 50% tall and 50% dwarf. Okay, that means you have what you call it as, you can see here, the 50% is like this and 50% is like this, which gives you a ratio which is 2:2. That is, 2 is capital T small t and the other 2 is small t, which can also be written as 1:1. This is the ratio. Here you cannot write a ratio here because here every plant is all the plant here is tall plant. Here you have tall plants here and dwarf plants here depending upon the phenotype. So you get a phenotypic ratio which is 1:1. Okay, phenotypic, you get a phenotypic ratio as well as the genotypic ratio which is 1:1. Here the phenotypic and genotypic will be different. Here the phenotypic ratio is only 1 because 100% is that it is 1, and genotypic ratio will be you have 2 of is to 2, which is you can take it as, because it is tall you cannot represent it in this particular form. Because everything is tall in that particular case, so it is 100% tall in that particular case, and here the ratio will be 1:1. So we saw how you get this 1:1 here. You cannot calculate 1:1 in this particular case because all the plants here, there is no phenotypic difference in this. As a result, you cannot calculate a phenotypic as well as the phenotype is all the same. As a result, it is 100% tall. And here you have two different phenotype, 50% tall, 50% dwarf. As a result, the phenotypic ratio will be 1:1. Is that clear? So this is the two back crosses. So back cross is nothing but when the F1 heterozygote is crossed with either parent, either with the dominant parent or the recessive parent. So if it is crossed with the dominant parent, you get all dominant or all tall in this case. In this case, you get half, 50% is to 50%, 50% tall and 50% dwarf. So this is what is called as a back cross. So if crossed with the dominant parent, the heterozygote is crossed with the dominant parent, all the resulting offsprings will have the dominant phenotype. If it is crossed with the recessive parent, half of the resulting offsprings will be having dominant phenotype and the other half will have the recessive phenotype. So this is what is called as back cross. And next we are moving on to the other one, which is test cross. So test cross is a type of back cross. Okay, and what is test cross in the sense is that when your heterozygote or F1 heterozygote is crossed with the recessive parent, such a cross is referred to as a test cross. That's why we said that test cross is a type of back cross. Okay, the monohybrid test cross we just mentioned in the earlier discussion of back cross, that is where we get 50% tall and 50% dwarf plants, was an example of a monohybrid test cross where you get the ratio 1:1. Okay, and coming to the other one, which is in a monohybrid test cross, half of the resulting offspring will have the dominant phenotype and the other half will have the recessive phenotype. And if you are moving on with a dihybrid test cross, what will happen is that you can see it clearly here. In a dihybrid, so this is an example of a dihybrid test cross. Okay. So in a dihybrid test cross also, so this was a heterozygote, a F1 heterozygote, which is crossed with the recessive parent of this. So you get the 4 gametes and also the gamete from this, and you get the different offsprings. 4 different offsprings are written here, and you can see that this is one phenotype is to 1:1:1. Every phenotype and every genotype is different, and this is they are having equal chances. That means the 25% chances for every offspring to be produced, and that is this is what is called as the dihybrid test cross. So in dihybrid test cross, this particular recessive parent is also called as a double recessive parent. In some references or some problems you come across such a term, double recessive. Double recessive is nothing but if you are taking a dihybrid cross, you have 2 characters and the factors which represent both the characters are recessive. Here BB is recessive, LL is recessive. There are 2 homozygous recessive in one plant, and that some authors refer to them as double recessive plants, or simply recessive also is okay. So test cross are usually conducted to confirm heterozygosity of the F1 individual. So we'll see how it is. So what happens is, if I am having a dominant, I'll take the previous example itself. I don't know the genotype of my plants, because when Mendel did the experiments, he just knew the phenotype of the plant. So now I'm having the phenotype of a plant which is tall. So tall can be either like this, or tall can be either like this. So I need to confirm whether my tall is this or this. So what I do is, you can do a test cross with both these possibilities. So this is the first possibility of a tall plant. This is the second possibility of a tall plant. In the first possibility, what I do is, I am going to cross it with the recessive parent. In the case what I am, you have the gametes, and what I get is capital T, small t, all are tall. 100% individuals are tall. So now what I do here is, I am going to cross this with this. So if I put a checkerboard, or I will just cross like the same. You have a T, small t here, cross a capital T and a small t here. So this is a small t. The offsprings that you get is capital T, capital T, capital T, small t. Sorry, you have only, I will make it more clear. I think the alphabets are not clear, so I put it once more. We'll divide it into two halves. This is one possibility, okay, and this is another possibility. So we are doing a test cross. I have a tall plant, and I need to know whether the tall plant is a homozygous plant or a heterozygous plant. So I test cross it with, or I am doing a test cross where I cross this plant with my plant in question with the recessive parent. So what happens here is, I have this here, this is the gametes, I get all tall plants here. So this is one possibility. And if I do like this, I have this small t and a small t here. So what I get is a capital T, a tall and a dwarf, okay, a tall and a dwarf, small t, and this is of the ratio 50%, 50%, or it is 1:1. And if you get such a type of phenotype in the after the test cross, so we do not know the genotype of the plant, we are just crossing, we are looking at the phenotype. I have a white plant. I consider it to be either a homozygous dominant or a heterozygous. If it is heterozygous, after the test cross, you will get a ratio 1:1. If you get a ratio 1:1, you can confirm that the tall plant which was in question is a heterozygous plant. And after a test cross, if you are getting completely the complete individuals as tall, you can confirm it as it is not a heterozygous, it can be homozygous. So heterozygosity can be confirmed using test cross. So test cross ratio will be obtained only if the F1 individuals is heterozygous. That means we can get either 1:1 in monohybrid cross and 1:1:1:1 in a dihybrid cross, only if the F1 individual is a heterozygote. If it is not heterozygote, you will not get this 1:1 ratio or 1:1:1:1 ratio. So dihybrid test cross is also used as a proof for the law of independent assortment. This is also considered to be the proof for law of independent assortment. It is because of law of independent assortment, because of that you get this 4 different alleles, and because of that you get 4 different here after crossing with a recessive parent. So this is also taken as an proof for the law of independent assortment. So I will explain why this is taken as a proof once more. So the proof is that here, because you have this 4 different possibilities, this 4 different possibility was what we explained as law of independent assortment, because you have 4 chances, and if you are crossing it with a particular recessive parent, you get 4 different type of individual in the ratio 1:1:1:1. If the independent assortment is not happening, you get only 2 type of gametes. As a result, the possibility after a test cross will be only 2 types of plants. Because I got in a dihybrid cross 4 different plants after 4 different varieties of plants after test cross, you can confirm the law of independent assortment. So this is what is called as a test cross. So coming to the next one, which is back cross. Back cross we discussed. Next is the last one, which is the reciprocal cross. So reciprocal cross is a cross which involves the same traits. Here we do not mix, or you do not change the characters. What you do here is you just reverse the sexes. Okay, in the sense of I will take this example, which is more simple. What you do here is, this is the first so this is the first experiment and this is the second one. So what you do here is you take we are considering two different two a monohybrid cross here. You have one type here and the other pair here. In this case, A1 is taken as female and A2 is taken as male, and you get a progeny like this. Okay, and in this particular case, you are just reversing the sexes. Okay, you can see here A2 is taken, here A2 was male, here you can see that A2 is now female and A1 is now male. So, and a cross is done. So such crosses where you just reverse the sexes from the first cross. In the first cross, if A is male, B is female, then in the second cross what you do is A will be taken as female and B will be taken as male. Such type of crosses are referred to as reciprocal crosses. So these are the 3 different types of crosses which we discussed now, which was back cross, then a test cross and a reciprocal cross. Thanks for hearing, and we'll discuss the other or the last session of this Mendelian genetics, which is the chromosomal theory of inheritance or chromosomal theory of heredity, in the next session.